of that they are used, but what they are was never mentioned. It was just a black box that was obviously easy to use because it wasn't worth mentioning. So this talk will be mainly, I'm working in a research lab, so it is really not like a core facility, so it will not be so much about big data management. It will be more about having a nice framework of processing data quite fast, because image processing seems to be a bottleneck, obviously, from the last talks we saw. And I want to show you one approach, this deep learning, which is quite fast and which hopefully makes it a little better so that we can process our data in speed that is reasonable and comparable maybe to the recording speed. Good. Good. Okay. So first of all, what kind of data sources we are going to process? What is possible with neural networks? Actually everything. So you can process from speech over spectral data, photographs, video. You can use microscopic data, 2D, 3D, over time, multi-channel, doesn't matter. And what are typical tasks? You already saw, for example, the registration task in the last talks about, for example, where you have the EMM image slices, which you have to register to each other. You have segmentation, especially in these neural tissues where you're interested in connectomics. This is quite crucial that is correct. And this is something the neural networks, at least my neural network, cannot do up to now perfectly. We have classification tasks, of course. We have different kinds of cells we want to classify. And first of all, we want to detect and segment them, or different tissue types. And overall, if we have 3D over time data, we of course also want to track. Good. You already saw a similar image. So that's from the EM segmentation challenge, which was set up by Ignacio. And we took part in this challenge in 2015 and were quite good then. Actually, there are some new results that are a little better now, but we're still in the top 10. And that's mainly the raw images, and that is the segmentation we get. So in one image, it looks quite good. If you look at the whole stack, well, it was not a well-aligned stack, so we had to align it manually. The connectomics is not really preserved. So we have to take care that if only one of these lines is broken, then everything is messed up. And neural networks are pixel-based, so they don't care whether this is the same as this. They just care if this pixel is correctly classified. What else do we have? Well, we can, of course, have many different data sources. For example, we have here just glands in histological sections, and we just want to segment these quite huge structures from the rest of the tissue. This can be easily done with neural networks. The problem here is mainly that the texture is quite clear, but these histological stains can be very different. So if you want to apply it to another sample, then you might have difficulties if they don't obey the same distribution of intensities here. Then, of course, you can have multi-class problems, not only two-class. So you can have, for example, this histological section, which contains 4 different cell types or 5 different cell types. Or let's say 5 classes. So we have bones, which are no cells. Then we have fat, and we have 3 interesting classes, which are, in this case, it's bone marrow tissue, and we're interested in megakaryocytes and their relation to surrounding progenitors for blood cells. What you can see here, you have some intermediate space, which is labeled as black. This is just cells we're not interested in, so they were not trained. Training is a problem. We will come to that later. Yeah, well, of course, we can have completely different data. That's really one of the bad examples where the network failed, although it looks quite nice. But you have to look at the goal of this. And if you're a dentist, you're probably interested in caries and not interested in whether you put a crown on some tooth. And the caries is here and was completely missed by this approach, just because it is a minority class, and so it was not trained properly. It would have required much more training data than was available. Then you cannot only segment. Sometimes you also want to detect something. In this case, you want to detect rosettes. This is a zebrafish primordium, so it's just forming these rosettes, which are deposited along the spine while the neural network is formed. And we are mainly interested just in these rosettes and their positions, and the network can also output just something like a heat map that tells you, okay, there is probably a rosette or not. And you can, of course, do that for other types of problems as well. Well, all I heard up to now was mainly processing 2D data. So we're in big data, and we want to also process 3D data, of course, especially if we have, for example, a SPIM that can produce very beautiful 3D data in masses. Then we also want networks that do 3D. And actually, we have an architecture that needs only very small adaptations, and we can do 3D with that. As you can see here, it is just kidney tissue of a mouse. And there you have these tubular structures, where you see there's black, which means it's internal of the tube. Then we have 2 membrane markers and the nuclear markers. So we have 3 channels here. And from these 3 channels, we can easily segment, first of all, the tubule itself, then the surrounding cells that form the tubule, and the rest of the tissue, which is shown in white here. And we can, of course, also have single cells, which looks quite simple, which might be easily possible to do it with elastic as well, I think. At least to detect these cells and to segment these cells. The difficulty here is we wanted to know which of them are alive, which of them are dead. And so dead cells are red, living cells are green. And in this case, we used time-lapse information. So if we have internal cell motion, then we can immediately see, okay, this cell has some internal cell motion, so it must be alive or it is not. And using the neural network, it can directly incorporate this without any hand tuning. Right, what you also can see, it is bright field data. So, well, segmentation in Z direction is not very nice. Actually, we can do better. We are just preparing a paper where we can show that we can even, from bright field data, get quite crisp boundaries, also in Z direction. And as a last example, it's another problem. It's, again, kidney tissue with a much further developed kidney. So actually, it's an adult kidney. And there you have these glomeruli you want to segment. So you want first to find them and then to segment them. And in this heat map presentation, you clearly see, okay, they are found. And if you find an appropriate threshold, usually it should be just the threshold of 0.5 or 0, depending on how you define it. Then you get a segmentation mask from that. Right. So we have lots of problems, and there are many, many more. And we are also processing many more. It's just a selection. And how do we approach it? Well, it's quite simple with one network. It's always the same network. I think some of you, at least one I know who knows it, probably many of you know it. It's the UNet. And the special things about the UNet, so I have just if you want to look it up, I have some references here. So that's the UNet in big. And what you see, it is just a neural network that takes as input an image or an image tile, just a small piece of an image. So with respect to the image sizes we saw, it's a very, very tiny tile of an image. It is processed by just filter operations, so it's just convolutions here. Then it's downsampled, convolved again, downsampled, and convolved again. So we get intermediate feature representations, which are then here stored on different levels of resolution. And these feature representation, from this, we try to regenerate another image. Actually, it doesn't matter what kind of image it is. In our case, it's mainly segmentation masks, so binary images or images containing some integer labels, not very much. You can, of course, also do something like regression with that. So you can also regress probability densities if you have a probability density label. But annotation of probabilities is quite hard. Good. So what does it do? It analyzes the image, encodes it in features, takes these features in the synthesis path, and synthesizes another image. That's basically it. And the good thing about it, it has millions of parameters, but you don't need to tune any of them. It is all tuned by itself just during the training process. There are maybe a handful of hyperparameters you need to know, so you have to adjust the learning rate and stuff like that. But there are standard settings that work most often, so I didn't change the standard settings for any of these examples. Good. What I didn't mention, we have these shortcut connections. So we pass the features that are computed in the analysis path directly to the synthesis path, that the network is able to reconstruct high resolution images from these data, and that it speeds up training a little. Well, when talking about training time, I must admit it trains for a while, depending on the data set. How complicated the data set is, it can take from days to weeks. So that's maybe one of the bottlenecks, but the good thing, training, you don't need to attend training, it trains. And at some point you will have a model that works, and if you train longer, it will work better, and at some point it saturates. So, what's the cool thing about this network? Well, actually, networks are pretty good in classifying. So there are classification networks around since, well, very popular ones since 2012. And they were able to take an image and produce one label. That's cool. But if we want to assume every pixel as one output of a network, then we have to process every pixel one after the other. That doesn't make much sense, because with the synthesis structure, we can directly, in parallel, compute the outputs for the whole yellow area here. And it incorporates information from this blue area, so you have a quite wide receptive field. So it can incorporate information from not only the pixel and its local surrounding, but from further away cell structures, for example. Good. So what is good about it? Well, we have a fully connected, fully convolutional architecture. So it doesn't matter what we input. It doesn't even matter what size of image we input. We can mainly, if we have enough processing power, if we have enough RAM, we can, in principle, input a full volume. If we don't, well, we can still input tiles, and the network will still train very well on these tiles. And therefore, actually, this network is able to learn from arbitrarily large data. And it can also classify arbitrarily large data then. It's only a matter of how many samples or how much time, how many tiles you have to process in the end. And you can easily parallelize it per tile. So if you have a cluster at hand, no problem. Just throw all the tiles on different cluster nodes and you have them back immediately, more or less. Right, so that's the cool thing about it. The uncool thing about it is that it is a learning approach, so you need training data. And we already talked about that. And I like the approach with only scribbling some of the structures you're interested in. And it's definitely a good idea to do that if it is possible, because it is really less effort. If you have problems that are not solvable by something like, okay, we put some scribbles or just put a threshold, for example, then the networks come into game. I would not try to solve every problem with networks. If it is too easy, do it another way, it is faster. But if it is really complicated and if you have a large data set, that's the other requirement. If you don't have a large data set and it is easy to annotate everything you have, then annotate it by manually in question. But if you want to process millions of images and want to maybe annotate only 10 or 12 or 20, then networks are what you want. So, actually, I talked about 10 to 20 images. In biology, this is sufficient. And you don't even need to annotate the whole images. But annotation must be pixel accurate if you want to get segmentation. And that's quite some effort. So if you tried to paint all cells in an image of 1024 by 1024, then you know that it takes time. And that's the reason why we want to get rid of annotating too much. Good. What can we use here? So we know the scales of our cells. We know the resolution of a microscope. We know there are no projective distortions compared to photographs, for example. We have no self-occlusion. We only have some absorption maybe, or cells may be next to each other or behind each other, but normally it's not so problematic. In EM, you don't have the problem at all. Then the absolute position and orientation of the structures is mostly irrelevant. If you have some cell culture, it doesn't matter whether a cell is orientated like that, or whether it's here or here. And your variability is moderate compared to natural images. If you have natural images, if I wanted to segment the whole audience here, I would have a problem because everyone looks completely different. You wear completely different clothing, and it's really a hard task to segment people compared to segmenting cells, because cells look mainly alike. So what do we do? Of course, if we know this, then we know, okay, shift and rotation is irrelevant, so we just shift our samples randomly. Then smooth shape variation is often irrelevant, therefore we can deform our images up to a degree, as long as they still obey the model we want to see. Then absolute brightness and contrast and noise are irrelevant, so we put just additional noise and change brightness and contrast and get millions of images from only 10 annotations, which we can use for training. Actually, arbitrarily many, because if we do this randomly, the network will never see the same image again. So this is how it looks like. If this is the original data set, you just crop some samples, rotate them randomly, flip them maybe, and apply some transformations, and that's it. Yes, I think I'm over time. Well, then I just wanted to show quickly, we can also do overlapping segmentations, and if you want to, you can compare it here. This is the ground truth, and this is our segmentation result. So it's really like, that's a problem because you normally can only give one label to every pixel of an image, and in this case, we lifted this restriction. I can tell you more later, maybe. Okay, so deep networks are extremely flexible. They can fit the learn image analysis pipeline from examples, only from examples. They allow classification, detection, segmentation, tracking. We have, with data augmentation, we can overcome the annotation bottleneck. Evaluation is extremely fast, within milliseconds for one of these tiles. But still, if you have a big data set, it can take some time. And you can, in principle, process arbitrary image sizes. So that's it. Thank you.